Hello, I'm Brenda Verhola from Aria from a Birdcage. Thanks for joining me for a look at sex-linked genes. By the end, I hope you have a good understanding of some of the mutated genes that are sex-linked, how they operate, and what they affect in a canary. Yes, I'm calling them mutated genes because that's what they are. The genetic code in the gene has mutated and become different than what is normal. It occurs in nature. It's spontaneous and it's a permanent change to the gene when it happens, and the creature that it happened to is different from others because of it. Today we'll be looking at mutated sex-linked genes, how they are trackable and predictable in offspring that are hatched from birds that have them. Here's where we start talking about some genetic stuff, but don't be scared. If you breed canaries, you're dealing with genetics, and learning some of these terms will be good for you. Each living thing, whether it's an animal, an insect, a fish, a flower, whatever, has chromosomes made of DNA that contain its unique genetic code. Many of these living things have one set of chromosomes that are sex chromosomes, and they determine if that thing is a male or a female. The sex chromosomes are referred to as X and Y, and this is known as the XY system. One set of a human's 23 pairs of chromosomes is their sex chromosomes. We can see that here in that a male is XY, he has one X chromosome and one Y chromosome in his pair, and a female is XX. Both of the sex chromosomes in her pair are X's. They get one chromosome from their mother and one from their father. In addition to controlling whether something is male or female, there are some genes on these sex chromosomes that affect other things. In humans, the genes for color blindness, hemophilia, and male pattern baldness are some of those. They are called sex-linked genes because they are a gene located on the sex chromosomes. In birds, things are different in that the male is XX, having two X chromosomes, and the female is XY, having one X chromosome and one Y chromosome. Truthfully, in birds, the ZW system is used. Males are ZZ and females are ZW. This ZW system also applies to some snakes, lizards, and insects. A different lettering system is used because biologists determined there is a difference in how the sex chromosomes work in these creatures as opposed to mammals and some other living things. So a different lettering system is used to represent that. Today we'll be using the XY labels for our talk because that's familiar to us. Also, many books and articles talk heavily about canary genetics, and they use the XY labels. I'm telling you about the ZW system so that if you talk with someone and they start mentioning Z's and W's, I want you to understand what it is and that is actually the correct way to refer to sex chromosomes in birds. So let's talk about birds. Birds have 40 pairs of chromosomes, with one of those pairs being the sex chromosomes. The Y sex chromosome carries only genes that determine and control femaleness. Only females have it. The X chromosome can have the particular mutated genes we're looking at today, specifically the ones that control certain colorations in the feathers. Because the female also has an X chromosome, that mutation can affect her too. So again, only the X chromosome carries mutations that we're looking at. So back to the pair of birds from the first slide. You'll notice that the male is shown with two X chromosomes and is bordered in blue. Blue for boy. The female is shown with the X and Y chromosomes that make her a female, and she is bordered in pink. Pink for girl. Also notice that the two X's in the male are different from each other. One is solid black and the other is striped. The male gets one of his X chromosomes from his father and one from his mother. The female's X has a checkerboard pattern to make it visually different from the X that our male has. Her X came from her father, and her Y came from her mother. We'll use these different looking X's throughout the presentation, and hopefully having them look different will help us understand the things we'll be talking about a little bit better. Here's our normal colored canary. We call this normal because there are no mutated or imported genes at work here. Our eyes see a green bird, but there's no green pigment in a canary's feathers. What we are seeing is an optical illusion. It's caused by three types of melanin being superimposed onto the ground color of the feather. Let's look at it this way. Here's a feather with a normal ground color, which is yellow lipochrome. The first of the three melanins is pheomelanin brown. It is concentrated on the feather edge and gets less dense toward the center. The second type of melanin is eumelanin brown, which lays on the feather concentrated down the middle with less density as it goes toward the edges. And the third type of melanin is eumelanin black, and like the brown, it's concentrated down the center and is less dense as it approaches the edges. Both the eumelanin black and the eumelanin brown also appear on the underfeather. You know, you have these types of melanins too. You don't have feathers, but your melanins control the color of your hair, your eyes, and your skin. Humans have eumelanin brown and eumelanin black and pheomelanin. I tell you this so you know these terms you're seeing here are not just for canaries. The sex-linked gene mutations we'll be looking at today all affect the melanins on the feather or the color of the ground color. That's why we're spending a bit of time to understand them in their normal state without any mutations. Here we're going to take a look at the normal melanins on the three ground colors of canaries. On the left we have the normal brown and black melanins. I put this on a grid background so you can see through it and see that it's transparent to some degree. When we look at the ground colors, we know that we have the normal yellow. We also have two genetic mutations that can cause the ground color to appear white. They don't color the feather white, they cause the absence of color. Think about that you can't mix colors of paint to create white paint. White is the absence of pigment. These mutations for white ground color are not sex-linked and they operate totally independently from the sex-linked genes we're looking at today. One of these mutations is called dominant white. The word dominant comes into play because what the mutated gene does is dominate the ground color pigment and push it almost entirely out of the feather. You'll only see traces of the ground color pigment on the ends of the flight feathers. The other mutation is recessive white, which completely masks all traces of the ground color and makes the feather appear pure white. It shows none of the ground color pigment. The third ground color is red, which is not a natural mutation of the canary gene. It was man-engineered into canaries by breeding in the black-hooded red siskin bird and then working to isolate just the gene for red feathers in the offspring. It's a gene that was transferred into canaries by hybridization between the siskin and the canary. I want you to know that the depth of color in the ground color of yellow or red depends on the genetic makeup of the bird. A bird with low lipochrome producing genes will not have the depth of color that a bird with high lipochrome producing genes has. You will see strikingly richer colors in yellows and reds on show benches where birds are being judged than you will see in song competitions because color breeders work hard to cultivate the lipochrome producing genes in their line. Color feeding plays a part, but the bird must have the right genetic makeup to achieve the quality and amount of pigment and the uniformity of color that we see it shows. We've already seen how the normal melanins affect the appearance of a yellow ground feather, making it appear green. If the ground color is white, the same normal black-brown melanins make the bird appear shades of gray, what we call blue. And if the ground color is red, the normal melanins give the bird a rich bronze color. This is a list of the X factor mutations that occur on the X chromosome. The first 5, brown, agate, Isabelle, pastel, and satinette, all affect the melanins. The last one, ivory, affects the lipochrome color, the ground color. Song breeders deal mostly with the brown mutation. We call it cinnamon, and if it appears on a white ground colored bird, we call it fawn. but it is possible that we could see any of these other mutations in our canaries because they can occur naturally. If you have a youngster appear in a nest that looks different, has a different feather color than what you're expecting, you may be seeing one of these other mutations. Color breeders have cultivated these, but I don't want anyone to think that if you're a song breeder that you can't see and enjoy these in your own birds. Let's look at each of these to get an understanding of what they do. The brown prevents eumelanin black. That's the black melanin that goes down the center of the feather, and all we see then are the brown melanins. Agate dilutes all melanins, so the ground color shows through more. The bird is brighter and there's more contrast between the ground color and the striping of the melanins. In Isabel, it combines both the brown and the agate mutations into one, so it prevents the eumelanin black and it dilutes the other two types of brown melanins. The pastel modifies the intensity of the eumelanin brown. That's the brown that goes down the middle of the feather. The satinette prevents eumelanin black and pheomelanin brown, which is the brown that lays on the edge of the feather. The only melanin showing in a satinette is eumelanin brown, and that's the brown that goes down the center of the feather and disperses as it goes toward the edge. The ivory mutation modifies the lipochrome shade of the feather, the ground color. Yellow appears as a pale lemon-colored feather. Red is modified to a shade of pink, and white still appears white, but in the dominant white, the yellow pigment that would be in the flight feathers disappears, and there's a pale suffusion of lipochrome that covers the whole body plumage. Imagine ivory on the keys of a piano. This is very difficult to see unless the white bird is color-fed. Okay, the last thing here is that a bird can have more than one of these mutations. It can be an agate and an ivory. It can be brown and pastel. It could be brown, pastel, and ivory. These mutations operate independently, and a bird can have one, none, or multiple of these mutations. We're going to study the brown mutation since it's most familiar to song breeders. Your understanding of how the cinnamon gene works will give you an understanding of all the sex-linked gene mutations. They can only appear on the X chromosome. They are all carried and transmitted to offspring in the same way, and they are all predictable in the same way. This slide shows 3 ground colors and the normal melanins on the left. The brown mutation prevents the eumelanin black from appearing, leaving just the 2 brown melanins, pheomelanin brown that tints the feather edge, and eumelanin brown that goes down the center and is on the underfeather. So the brown mutation on the yellow feather gives us that warm cinnamon color. On the white ground color, we see the fawn color, a cool beige. And here it is on the red ground color. Color breeders describe their colors by saying the ground color first and then the mutation. So this would be a red-brown in their lingo. I will also tell you that, just like in the ground colors, there are varying degrees of quality of the melanin-producing genes. Not all browns are created equal. If having the best color in your cinnamon or fawn birds is important to you, you need to be attentive to the depth of color in the melanins that your parent birds are contributing. The brown mutation also changes the eye color to red instead of black. Notice the chick on the left has garnet-colored eyes, while the one on the right has the normal black eye. This is your first clue that the chick has the brown gene, even before it feathers out. On the day it hatches, the eyes of the chick appear to be that garnet color, and they darken as the bird grows to where you can hardly see this when the bird's an adult. Here we have our 2 birds, the normal male and the normal female. As we've said before, the sex-linked gene can only appear on the X chromosome, so let's look at that. A male can have the mutated gene on one of his X's or on both of them. I'm using the M to indicate the X factor mutated gene. I could use B for brown, or I for ivory, or A for agate, but because this slide is an overview where we're talking about any of the sex-linked genes, I'm labeling it M as a catch-all for mutation. This guy can have his M gene on either of his X's, and if he has it on either one of them, he would be a carrier of that gene. He will look completely normal in appearance. He would be a green bird. When a male has the mutated gene on both of his X's, meaning that the X he got from his father had it, and the X he got from his mother had it, he shows it in his coloring. I've not shown any particular color here because we're illustrating any of the mutations, but I've shown it as different than normal because this bird will look different. Any of the melanin affecting sex-linked genes will show themselves wherever this bird has melanin. If it's the ivory mutation, wherever he shows his ground color, we will see the ground color change that gene causes. Now for females. When a female has the mutated gene on her X, she will show it in her coloring. This looks like we're going to play tic-tac-toe, but this diagram is actually a Punnett square. It's named after the guy who came up with this way to help biologists determine the probability of offspring from two specific parents having a particular genetic trait. It's been used to predict all kinds of things, from eye color to whether someone will be colorblind to whether pea pods will be yellow or green. The Punnett square works like this. We determine the pairing we want to make and what genetic trait we are evaluating. We'll say that this male has the brown mutation on one of his X chromosomes, making him a carrier. I've used the small letter B to indicate which X is carrying the gene for brown. This guy is a green bird. He looks completely normal. He has all the melanins of a normal bird. Our hen is also normal. To set up the chart, we put the male's 2 chromosomes across the top of the chart and the female's 2 chromosomes down the left side. Next, we'll drop the male's information into the square below each of his X's. Just copy down what's at the top and place them in the space below. Then, we'll copy the female's information on each row into the squares to their right. So the checkerboard X goes to both squares to the right of it, and the Y goes to both squares to the right of it. There are 4 possibilities of outcome of this pairing. The top left square has the mutated X from the father and the normal X from the mother. The top right square has the normal X from the father and the normal X from the mother. Either of these would be males because of the double X's. And the one on the left would be a carrier of the brown mutation because he has the brown gene on only one of his X's. For the bottom squares, we see the one on the left has the brown gene from the carrier father and the Y from the mother. The square on the right has the normal X gene from the father and the Y from the mother. Either of these would be females because of the XY combination, but the one on the left would look different. She would be a cinnamon bird. She has the mutation on her only X, and she will show it to you in her eyes and anywhere that melanin appears in her feathers. So the Punnett square for this pairing tells us that the offspring can be carrier males, normal males, brown females, or normal females. Sometimes the first time we discover that we have a sex-linked gene mutation in our birds is when we notice a chick in the nest that's feathering out a different color than what we thought it would be. And we wonder, where did that chick come from? In this case, the parents look normal because they're all green birds with normal melanins. And a chick appears in the nest that's not normal. Sometimes the first time you know a male is a carrier of brown is when he fathers a chick in the nest that's brown. If this happens, mark the records for the male so that you know that for future pairings. We also know that the brown chick in this nest is a female. The one X it inherited has the brown gene, and she's showing it to us in her feathers. She will show it even before she feathers out because we'd notice her red eyes. So if the mutation were ivory, We would see the same possibilities if we had a pair that looked normal, but where the male was a carrier of ivory. I've used a small letter I to show that ivory gene. If a chick in the nest had a paler version of the ground color than her parents, we know it's ivory, and we would know that the father is a carrier of ivory. We would also know that the chick is a female. So again, the chick that's in the lower left-hand square has a lighter shade of yellow than every other bird possibility in the nest, and than that of her parents. So we know that's that ivory mutation showing it to us. We'd really have to wait until she feathered out as an adult to evaluate her color, because as a chick they tend to have muted colors and they get that vibrancy in their adult feathers. So don't make the judgment too soon, but know that if she's different than the parents, that is most likely that ivory gene. The four possibilities that the Punnett square shows us are scientific possibilities, but each chick is a throw of the dice, or a spin of the wheel in this case. We know the four possibilities, but the genetic combination for each egg that's laid in a nest is determined by which chromosome from the dad and which chromosome from the mom were there in that magic moment when the egg was fertilized. The wheel spins and the first egg is fertilized and joins the normal X chromosome from the dad and the normal X chromosome from the mom, and we have a normal male develop. The wheel spins again for the next egg, and the X chromosome with the brown gene from the dad joins with the Y chromosome from the mom, and we get a cinnamon female. The wheel spins again for the next egg, and a normal X chromosome from the dad is joined with a normal X chromosome from the mom, and we have another normal male. So in the nest, we'd see a red-eyed cinnamon chick and two normal chicks. We'd know that the red-eyed chick was a girl. When they matured and we determined that the normal chicks were boys, we should mark their records as possible cinnamon carriers, because we can't tell by looking whether they are or not. Not until they father their own young chicks will we know. And again, it may take several nests, because the first season you breed them, they may not produce that cinnamon hen that would be your confirmation. Let's look at another Punnett square using a brown male and a brown female. Again, I'm using the small b to indicate the presence of the brown mutated gene. Both X's of the male shows the B, and the X of the female shows the B. And both birds show us in their feather color that they are brown, wherever they have melanin. Just as before, the male's information goes across the top, and the female's information goes down the left side. You drop the male's information down into the squares beneath them, and copy the female's information across to the squares on their rows. You can see that all of the offspring will have the B mutation on their X chromosomes, and all will show us that they have it wherever they have melanin. Now, I've been careful to say that these birds would all show us that they have the brown mutation wherever they have melanin. I set up a pair like this once. One of the parents was a cinnamon bird, and the other was a lightly variegated cinnamon bird. One of the chicks feathered out to look like this, a totally clear yellow bird. Would this bird be a cinnamon bird? Yes, it would be. We know that genetically it is a cinnamon bird, even though it's not showing us any melanin. We know this because its parents cannot make any offspring that's not genetically a cinnamon. What is at play here are the genes for variegation. Those are called variegation factor genes, and they are independent of the X factor genes. Variegation genes determine the degree of variegation on a bird. In song canaries, we see a lot of variegation, which I love. Clear birds like this one are produced when a mutation which suppresses the formation of melanin is taking control. This bird has no melanin being expressed because of its variegation factor genes, but it does have the B factor on its X chromosomes. Genetically, it is a cinnamon bird. This is where keeping records on your birds you breed and what they produce is essential. For this bird, I marked its records, but I also put a brown plastic band on its leg so I'd be reminded when looking at it in the cage that it was a cinnamon. So for any of the sex-linked genes that affect melanin, if your bird doesn't show you any melanin because of its variegation genes, your records are important. Now we aren't going to go through a Punnett square for each combination of male and hens with a sex-linked mutation, but I want to show you how you can set up a pairing chart of your own using the Punnett square concept. Here across the top of the chart, I have put the three variations of males, the normal, the carrier, and the brown. Down the left side, I have put two types of females, the normal and the brown. Again, we're just looking at the brown or cinnamon mutation, but all of these X factor mutations would work the same. If I pick the brown female and the brown male, all I need to do is look at the intersecting square to see what the possibilities in the nest would be. I can get brown males and brown females. Every chick in the nest would look like and be a cinnamon bird, with the exception of that variegation factor, but we would know genetically every bird in the nest would be cinnamon. If I pick the normal female and the carrier male, again I just look at the intersecting squares and there are my possibilities. Normal males, carrier males, normal females, and brown females. Here's another way to slice it and add a bit more to the complexity. This pairing chart adds in the white ground color. Across the top, I have all the possibilities of male: green, blue, green carrier, blue carrier, cinnamon, and fawn. Down the left side, I show females: green, blue, cinnamon, and fawn. The first footnote at the bottom tells us that any chick with the asterisk beside it will have garnet-colored eyes. The second footnote reminds us that green in this chart is a yellow ground bird with normal melanins, and a blue in this chart is a dominant white bird with normal melanins. In the intersecting squares of a particular male with a particular female are the possible results of that pairing. I need to tell you that pairing two dominant white ground color birds together is not recommended. So here I'm blocking the white ground to white ground possibilities out. We're not studying the genetics of ground colors today, and this gene is not sex-linked. But there's a lethal factor that will affect the particular chick whose spin of the wheel gives them the dominant white gene from both of their parents. The possibility of that is 25%. But we know that each fertilized egg gets its own spin of the wheel. The spinner could land 3 times on this possibility for the eggs in the nest, and all those chicks would die. It could land on other possibilities instead, and all those chicks would be fine. If the ground color is recessive white, the gene works differently, and the pairings between those birds are fine. But I didn't do that for this chart, because then we'd have to add in the complexity of carriers of recessive white. Again, that gene works differently than the dominant white. I posted this chart on the website, and you're welcome to print a copy of your own. Now we're going to leave behind the Punnett square and charts and look at pictures. This is the fun part. I'll show you the best examples I could find of most of the sex-linked mutations on various ground colors. I don't have all of them, but I have enough to help you recognize them when you see them. Here the bird on the left is a normal green canary, yellow ground color and normal melanins. Most of my canary examples in this section are colorbreds because I could find the best examples of colors with them. But my example here is a border fancy. Borders are known for their high quality color, and this guy is a great example of that. You can see the striations of eumelanins that go down the center of the feathers and the uniformity of the melanin coverage. He appears as a beautiful olive green. The canary on the right shows us the brown mutation, or cinnamon as many of us call it. The eumelanin black is gone and only the brown melanins are being expressed. Here on the left we have a blue canary, the normal melanins on a white ground color. The shades of gray have a brown undertone from the brown melanins. The bird on the right shows us the brown or fawn mutation on a white ground color. The black is gone, and we see just the brown melanins. An agate mutation is shown here. Our normal green is on the left, and a bird with the agate mutation is on the right. Agate dilutes all melanins. The eumelanins still have the greatest concentration down the center of the feather, but there's less melanin dispersing outward, so more ground color is visible. This makes for a stronger contrast. I also want you to notice his Fu Manchu mustache, which is a trademark of the agates. Here on the left we see a normal blue, and on the right we have the agate mutation on the white ground color. The Fu Manchu mustache is there, and the melanins are showing good contrast with the white ground color. That's more visible because of the dilution of the melanins. And here's the agate mutation on the red ground color, again with the normal melanins on the bird to the left and the mutation on the right. This example shows the diluted eumelanin black is more of a charcoal color compared to the black on the bird with the normal melanins. And there we can see his Fu Manchu mustache. Here's the Isabel mutation on the right, and it's a nice example for us. Isabel combines the brown mutation and the agate mutation in one. So brown removes the black melanin completely, and agate dilutes all melanins. What we see is more of the yellow ground color because of that dilution, and we just see brown melanins. We'll see a trace of that Fu Manchu mustache on the agate, but because we don't have the black melanin, it's not as apparent. Here's the white ground color bird with the Isabel mutation. There's no eumelanin black, and all the brown melanins are diluted. This makes for a beautiful bird. And again, we see just a trace of the Fu Manchu. Satinette prevents the eumelanin black that runs down the center of the feather. It also prevents pheomelanin brown, which is at the feather edge, and the only pigment that remains is the eumelanin brown that goes down the center and is on the underfeather. Satinette birds have red eyes that don't darken as the bird grows older like they do in cinnamon. So the red eye in that picture is really that bird's eye. It's not just a bad photograph. You can see a very subtle brown striping of melanin here in its feathers. One sex-linked mutation, and look at the huge difference when comparing it to the normal bird on the left. Again, this is a naturally occurring mutation that could spontaneously happen in your bird room. If a chick in one of your nests feathers out to look like that, you would notice it, and wouldn't that be neat? This is the satinette mutation on a white ground color bird. No eumelanin black or pheomelanin brown, just the eumelanin brown that's concentrated down the center of the feather and is less dense as it goes out from the center. It shows up better on the white ground bird because there's no pigment in the white feather to distract from it. And this is the satinette mutation on a red ground color bird. Like the yellow ground bird, the pigment of the ground color gives that eumelanin brown a colored canvas, so the striping is much more subtle than what we saw on the white ground color bird. But it's there. The ivory mutation is the last sex-linked mutation, and it doesn't affect the melanins at all. It affects the ground color pigment. The bird on the left is a yellow intensive bird, and the bird on the right is a yellow ivory bird. You can see the change of color from one bird to the other. It's a different hue, a shift of the color. Keep in mind that these photos are of show quality color-bred birds. Their breeders have in their breeding program a focus on strong lipochrome producing genes. When I look at the ivory bird on the right, the yellow canaries I have in my American Singer flight are more of this color than they are the bird on the left. That's because in my bird room, the depth of color doesn't matter. What I care about is the song of the bird. But if I had an ivory mutation in my bird room, it would be noticeable. The chick would feather out to be a different color than its parents, an obvious lighter shade of yellow. And I would love it if that would happen. And this is the ivory mutation on the red ground color canary. we see the change in color that the ivory causes. The red ivory bird is one of my absolute favorite colorbreeds. And this last slide shows an example of a bird that has two of the sex-linked gene mutations. Remember that I told you that a bird can have more than one mutation? The bird on the left is our normal blue, a white-ground colored bird with black and brown melanins. The bird on the right has two mutations. Can you guess what they are? Well, it doesn't have any black melanin, so that means it has that brown mutation. That's one. The other mutation is pastel, that modifies the intensity of the melanin brown. That's the one that's concentrated down the center of the feather. With the black eliminated and the brown modified in its intensity, this bird has almost no striping or markings. The reduction in melanin intensity caused by the pastel makes it appear as a paler brown. I hope this information helps you understand cinnamon and other sex-linked genes that may be at play in your bird room. I'd love for you to let me know if it was a help to you or if there are other topics you'd like to see covered. Enjoy your day, and again, thank you for joining me.